Welcome to the Deep Dive. We're the show that takes a complex topic, digs into the sources, and boils it all down so you can get up to speed without getting overwhelmed.
And today we're tackling something huge, something, well, universal, aging. It's fascinated people forever, hasn't it?
Absolutely. But for so long, how our bodies change over time just felt like this big mystery.
Right. Incredibly complex. But things have really shifted. Scientists now have this amazing framework, almost like a Rosetta Stone, you could say, to actually make sense of it all.
The hallmarks of aging.
Exactly.
And that's our mission today in this deep dive. We're going to unpack that framework.
Yeah.
We'll look at how all these different biological changes add up to aging. And here's the really mind-bending part.
Yeah.
How some processes that are actually good for us when we're young can kind of flip and turn against us later.
That double-edged sword idea, it's central to this.
We're going to explore that whole intricate dance, pulling insights from the research you've shared. Think of it as a shortcut for you, the listener, into understanding what's really going on at the cellular level.
Okay, so this framework, the hallmarks of aging, it first came out in 2013, really giving researchers a way to organize this incredibly tangled web of factors in aging.
Right.
And it basically groups these changes into three main buckets, like different types of wear and tear, maybe.
Okay, 3 buckets. So let's start at the beginning, the really fundamental stuff, the changes that are just bad news from the start and pile up over time, the unavoidable damage maybe. What are these primary hallmarks?
Great question. These primary hallmarks are exactly the sort of foundational damage that accumulates. You've got genomic instability, basically, our DNA getting errors, damage.
Yeah, okay.
Then telomere attrition, those protective caps on our chromosomes, they get shorter. It's like a cellular clock ticking down.
Right, the caps.
Then epigenetic alterations. These are changes in how genes get read, how they're expressed, without changing the actual DNA code itself.
So changes to the instruction manual's notes, not the text itself.
Yeah. Good analogy. And finally, loss of proteostasis. Think of your cells like little factories making proteins, right? Proteostasis is the quality control.
Making sure the proteins are folded right, getting rid of bad ones.
Exactly. And as we age, that system gets sluggish, bad proteins build up, gum up the works, the factory gets less efficient.
That factory analogy really helps picture it. Okay, so that's primary. What about the next category, the antagonistic hallmarks? You mentioned this is where that paradox comes in, protective early on, but harmful later. What makes them so sneaky?
They're fascinating, really, because they perfectly show this idea of antagonistic pleiotropy. It's this evolutionary trade-off. Something helps you survive and reproduce when you're young.
Vital, then.
But it has downsides later in life. So here we find things like deregulated nutrient sensing, cells get confused about how much fuel is around, then mitochondrial dysfunction, problems with the cell's powerhouses. Big one.
The energy factories.
And another huge one we'll definitely dig into more, cellular senescence. Cells stop dividing, they become these sort of zombie cells.
Zombie cells, I like that.
Yep.
Okay, and the third category, integrative hallmarks. How do they fit?
These really capture the bigger picture, the body's overall ability to maintain balance, to repair itself, tissue homeostasis.
Right, keeping things stable.
So here you see stem cell exhaustion. Our body's amazing repair crews just get depleted over time.
Fewer reserves.
And altered intercellular communication. Basically, cells just stop talking to each other effectively. Communication breaks down, coordination fails.
So it sounds like a comprehensive list, but you mentioned it's not set in stone. Science keeps refining this.
Oh, absolutely. It's a living framework. Just recently, they did a major update and added 3 new hallmarks.
Really? What are they?
Well, disabled macroautophagy, that's the cell's main cleanup crew for recycling damaged parts that actually got moved up, added to the primary hallmarks now. It's seen as that fundamental.
Wow.
And then 2 more join the integrative category, chronic inflammation, which people often call inflammaging.
Heard that term?
And dysbiosis. That's an imbalance in our gut microbes, our microbiota. Huge impact.
Okay, so having this whole framework, these categories, why is that so important for the science? What does it actually let researchers do?
It's like giving them a shared map, a scaffold for all aging research. It helps design better experiments, make sense of complicated data.
Right.
And maybe the coolest part, it helps us understand longevity in other species. Like, how do bowhead whales live for 200 years?
The longest lived mammal, yeah.
Or giant tortoises, or even that immortal jellyfish, Turritopsis dohrnii.
The one that can revert back to a younger state.
Exactly. This framework helps pinpoint the biological traits that control lifespan across the board. It lets us ask, what are they doing right at the cellular level?
That's a really powerful lens. Okay, let's zoom in, like you suggested, on one of those antagonistic ones. Cellular senescence. The zombie cells, it's such a striking term. Why is this such a double-edged sword?
It really is the perfect description. Senescence is when cells just stop dividing. Permanent growth arrest. But they don't die. They stick around, still metabolically active.
Okay.
And their original job, their good job, is crucial. Stopping damaged cells from multiplying endlessly. It's a built-in anti-cancer mechanism.
So a safeguard.
Definitely. And you can actually see them change. They get bigger, flatter, look kind of bloated and sluggish compared to healthy cells.
And there is a good side, isn't there?
Yeah.
People forget that sometimes. It's not all bad, especially early on.
Not at all. Senescence is vital for things like embryonic development, shaping tissues as we grow. It's critical for normal wound healing. Certain cells become senescent to help close a wound properly.
Like fibroblasts, right?
Exactly. And like we said, it's a powerful tumor suppressor. It even helps, surprisingly, in the early stages of acute kidney injury. If you block it too early, then the kidney might not recover properly.
But then comes the dark side. Why does it become such a menace as we age?
Because these cells, designed to be protective, they don't just sit there quietly once they stop dividing. They start spewing out this inflammatory cocktail of molecules.
The SASP, senescence-associated secretory phenotype.
That's the one, the SASP. And this cocktail, shockingly, can actually create an environment that helps tumors grow later on. It also fuels chronic inflammation throughout the body.
So the guardian becomes the arsonist, almost, like that fire alarm that won't shut off and eventually helps burn the place down.
That's a great way to put it. They're not just taking up space, they're actively, well, poisoning the local environment.
And it goes way beyond just cancer risk, doesn't it?
Oh, absolutely. Senescence is seen as a key driver of aging itself, and it contributes to a whole laundry list of age-related diseases: osteoporosis, metabolic syndrome, type 2 diabetes, atherosclerosis, neurodegeneration,
Glaucoma, chronic kidney disease.
you name it. And remember, inflammaging, that chronic, low-level fire. Senescent cells pouring out SASP are a major fuel source for that. Plus, they contribute to immune system decline, stem cell exhaustion. It's a cascade.
Wow. So what pushes a cell into this zombie state? What are the triggers?
It's a mix of things, really. Major triggers include significant DNA damage from radiation, chemicals, or just a wear and tear of metabolism creating those reactive oxygen species, ROS.
Okay, DNA damage.
Telomere shortening, when those caps get too short, that's another big one. Also, if genes that normally control cell growth get messed up, like oncogenes getting activated or tumor suppressors getting lost.
So genetic and chromosomal issues?
Right. But also epigenetic changes, damage to organelles inside the cell, even things like heat shock, metabolic stress like high glucose levels, or just physical damage can push a cell into senescence.
Given how central they seem to be, can scientists actually find these senescent cells in the body? Identify them?
They can, yes, but it's tricky. There are biomarkers, things like SA-ball, P16, P21, that are often found in senescent cells, but none of them are perfectly specific or sensitive on their own. A healthy cell might show one marker under certain conditions, for instance. So finding better, maybe non-invasive ways to reliably detect only senescent cells in living tissue, that's still a major goal in the field.
Makes sense. Okay, let's pivot to another huge hallmark, mitochondrial dysfunction, the cell's powerhouses. Crucial for energy, but again, there's a paradox here too, isn't there?
There is. As we get older, there's a very clear decline in both the quality and the activity of mitochondria in many tissues.
And that contributes directly to
age-related functional decline. Think muscle loss, sarcopenia. That's tightly linked to mitochondria not working as well.
And this is where that antagonistic pleiotropy concept really bites again. Mitochondria give us the energy for youth, for reproduction, for everything.
Absolutely essential.
But their gradual decline contributes to frailty later on. The very engine that drives us can also wear down.
Exactly. And it gets even weirder with this idea called mitohormesis.
Hormesis, meaning a little bit of stress is good.
Pretty much. In some organisms, a slight impairment in mitochondrial function can actually trigger protective stress responses, like the mitochondrial unfolded protein response, the UPRMT. And this can, paradoxically, extend lifespan.
Wow. So perfect mitochondrial health isn't always the goal. A little nudge might activate defenses.
It seems that way in some contexts. It's like the cell senses a small problem and overcompensates, boosting its overall resilience.
Fascinating. But then there's the ROS issue, reactive oxygen species, friend or foe. We hear they cause damage.
They definitely can. Mitochondrial dysfunction is a major source of excess ROS, and that excess ROS can damage DNA, proteins, lipids, contribute to senescence.
Rusting from within idea.
Right. But ROS aren't all bad. They also act as important signaling molecules within the cell. Some level is necessary. And get this, in some studies, increasing ROS has actually increased lifespan.
How's that work?
Again, it likely ties back to hormesis. A controlled pulse of ROS might trigger protective mechanisms. It's all about balance. Too much is clearly damaging, but eliminating them entirely isn't the answer either. It's this dynamic equilibrium that gets harder to maintain with age.
Okay, so what specifically goes wrong inside the mitochondria as we age? What are the mechanisms of dysfunction?
Several key things. One is mutations accumulating in the mitochondrial DNA itself. Mitochondria have their own little genome.
Right, the mtDNA.
Those mutations build up. Then there's problems making new mitochondria. That's called mitochondrial biogenesis. That process can become impaired.
So fewer fresh power plants being built.
Exactly. Also, changes in the levels of crucial metabolic molecules that mitochondria handle can mess things up. And a really big one is problems with mitophagy.
The cleanup crew, again, specifically for mitochondria.
Yes. Mitophagy is how the cell gets rid of old, damaged mitochondria. If that process fails, the junk just accumulates, like letting broken down cars pile up.
And that's linked to diseases.
Definitely. Mutations in genes controlling mitophagy, like PNK1 and Parkin, are directly linked to Parkinson's disease. It highlights how crucial this cleanup is.
So for you listening, why care about your mitochondria? Well, their health impacts everything from your energy levels to your muscle strength. And the good news, things like exercise are potent stimulators of mitochondrial biogenesis.
Absolutely. It's a clear link between lifestyle and cellular energy production. Exercise really can help counteract that age-related decline.
Okay, we touched on this with senescent cells, but let's focus now on inflammaging, that silent fire. This really speaks to aging as a systemic thing, affecting the whole body, right?
It does. Inflammaging is this chronic, low-grade inflammation that creeps up with age, even when there's no obvious infection or injury. It was a revolutionary idea when Professor Franceschi introduced it back in 2000.
Changed the game.
Completely. It framed aging not just as isolated decay, but as this simmering, body-wide inflammatory state.
And again, that dual nature. It starts as adaptive, like the immune system keeping a record of battles fought.
Sort of, yeah. It can be seen as the result of a lifetime of exposure to various stressors and pathogens, your immunobiography, as some call it. Your immune system learns and adapts.
What the?
But then, if that system stays dialed up too high for too long, or crosses some threshold with age, it becomes maladaptive. It fuels pathological aging and many age-related diseases.
And the consequences are serious. It's a major risk factor for poor health outcomes in older adults.
Huge risk factor for morbidity and mortality. And here's a wild connection back to mitochondria.
Uh-oh. How did it fit in here?
Because mitochondria evolved from bacteria millions of years ago, when they get damaged and break open inside our cells, bits of them look foreign to our immune system.
Like bacterial debris.
Exactly. Things like their DNA, which has features similar to bacterial DNA, or certain protein fragments. These act as DAMPs, damage-associated molecular patterns.
Alarm signals.
Right. And our cells have internal alarm systems, like the NLRP3 inflammasome or the EST-King pathway, that detect these mitochondrial bits and trigger inflammation. So damaged mitochondria can directly fuel inflammaging.
Wow, so the cell's own power plants can trigger an immune alert if they break down. But they aren't the only source fueling this fire, are they?
No, not at all. We already talked about senescent cells and their SSP constantly pumping out inflammatory signals. That's a huge contributor.
Right.
And then there's dysbiosis, an unhealthy balance of microbes, especially in the gut. An inflamed gut can leak bacterial products into the bloodstream, constantly poking the immune system.
The gut connection again, it's everywhere.
It really is. And there's also this idea of trained innate immunity. Your first responder immune cells can get trained by past infections to react more strongly.
Which sounds good.
Good against repeat infections? Maybe. But it can also lead to them being hyper-reactive later in life, or even becoming paralyzed and unable to shut down properly after being overstimulated. It all feeds into that chronic, smoldering inflammation.
Understanding this, like you said, was critical during COVID-19 for older people, where that inability to turn off inflammation was so dangerous.
Precisely. It highlighted how crucial managing inflammaging is for resilience.
Okay, let's shift gears to telomere attrition, the chromosomal clock. We mentioned these protective caps, TTG repeats at the ends of chromosomes. They sound simple, but they're critical.
Absolutely fundamental. Because of how DNA replication works, the end replication, a little bit of the telomere is lost each time most cells divide.
So they get shorter and shorter.
Right. Acting as this biological clock, it limits how many times a cell can divide, and when they get critically short, it signals danger, potentially leading to genomic instability or senescence.
So how does the body counteract this shortening, at least in some cells?
With an enzyme called telomerase. Its job is to add those TTTG repeats back onto the ends, lengthening the telomeres.
But it's not active everywhere, is it?
No, that's the key. It's highly active in stem cells, which need to divide a lot throughout life, and in germ cells. But most of our regular body cells, somatic cells, have very little telomerase activity, so their telomeres just progressively shorten.
Which leads to that critical cancer paradox.
Mm.
Shortening is bad for cell longevity, but good for preventing cancer.
Exactly. It's a natural brake on uncontrolled cell division. If a normal cell starts dividing like crazy, it will eventually hit this telomere limit and stop, preventing a tumor from forming.
But cancer finds a way around it.
In the vast majority of cases, yes. About 85% of cancers manage to switch telomerase back on. This allows them to maintain their telomeres, overcome that limit, and achieve replicative immortality, a key hallmark of cancer.
It's always walking that tightrope. Now, besides telomerase, there's also the shelterin complex. What does that do?
Shelterin is a group of proteins that binds specifically to the telomere DNA. Think of it like a protective helmet for the telomere cap.
Protecting the protector.
Sort of. It prevents the cell's DNA damage machinery from mistakenly seeing the chromosome end as a break that needs repairing. If shelterin fails, the telomere becomes uncapped, unstable, and confused with other chromosomes, leading to genomic chaos.
So telomeres shorten with division. What else makes them shorten faster?
Other stressors definitely accelerate it. Oxidative stress is a big one. Those ROS molecules we talked about. Telomeres are quite vulnerable to them because of their specific DNA sequence.
Okay, oxidative stress.
And chronic inflammation. Remember inflammaging. Those inflammatory signals, like certain cytokines, can actually suppress the activity of telomerase, making the shortening worse. It creates a vicious cycle. Inflammation shortens telomeres. Shortened telomeres can trigger senescence. Senescent cells produce more inflammation.
They all feed each other. Which brings us to the telomere mitochondrial axis. Sounds like another critical feedback loop.
It really is. It highlights how interconnected these hallmarks are. Basically, dysfunctional telomeres, maybe they're too short, or uncapped, can signal stress that leads to increased ROS production and mitochondrial dysfunction.
Okay.
And conversely, mitochondrial dysfunction, producing excess ROS, can directly damage telomeres and accelerate their shortening. It's a downward spiral linking chromosome ends to cellular energy production and damage.
So for you, the listener, what's the takeaway? Telomere length is being looked at as a potential biomarker of biological age, reflecting accumulated stress. Its integrity seems deeply tied to overall cellular health.
Definitely a hot area of research.
All right, so we've explored these intricate, often paradoxical hallmarks. The big question then becomes, what can we do about it? The goal isn't just living longer. It's extending health span, right? More years spent healthy and active.
That's absolutely the focus. Extending lifespan without health span isn't really the goal for most people or for the science. It's about quality of life.
So what are the promising strategies emerging from all this research? How can we target these hallmarks?
Well, there's a lot happening. One major area is tackling those zombie cells with senolytics and xenomorphics.
Okay, senolytics. Lysis, meaning to destroy.
Exactly. Senolytics are drugs designed to selectively find and eliminate senescent cells. Xenomorphics are a bit different. They aim to change the behavior of senescent cells, maybe suppress that harmful SASP, without necessarily killing them. Drugs like metformin or rapamycin might have some xenomorphic effects.
Are these being used now?
Clinical trials are definitely underway for several senolytic compounds. It's promising, but still early days in terms of widespread use and long-term effects. More research needed, for sure.
What else?
Stem cell therapies are another avenue. The idea is to use stem cells, or even the little packages they release called extracellular vesicles, to help repair tissues and potentially reduce the burden of senescence.
Sounds futuristic.
It is, but it's happening. Though the sources rightly point out that the effects can be very dependent on the type of stem cell, where it's put, the specific context. It's not a simple fix.
And then there are things maybe more within our reach.
Absolutely. Lifestyle interventions. The impact of things like regular exercise and caloric restriction or dietary patterns mimicking it is huge.
How do they help?
They hit multiple hallmarks that can improve mitochondrial function, reduce inflammation, potentially limit SASP, influence epigenetics. Diet also plays a massive role through epigenetics and shaping your gut microbiota.
So diet and exercise remain foundational.
Absolutely critical, yes.
What about targeting telomeres directly?
That's being explored too, but it's tricky. There are telomerase activators compounds like cyclooestrogenol found in some supplements aiming to lengthen telomeres.
But the cancer risk.
That's the huge caveat. If you allow cells to bypass the telomere clock indefinitely, you risk promoting cancer. It's that paradox again, a very delicate balance.
Are there other approaches?
More experimentally, yes. Telomerase gene therapy has shown promise in mice using a harmless virus to deliver the telomerase gene extended lifespan and reduced aging signs without increasing cancer in those models.
But in humans,
Huge safety and ethical hurdles for direct gene therapy in humans right now. It's complex. Interestingly, though, studies on mice engineered to have hyper-long telomeres from birth showed some benefits: less DNA damage, better metabolism, even less cancer.
less cancer with longer telomeres. That seems counterintuitive.
It does, but it suggests that maybe extreme length provides extra stability or buffering capacity. It implies that safely modulating telomere length, if we could figure out how, might be beneficial. It's an ongoing puzzle.
And for mitochondria, interventions there.
Yes, things like trying to boost levels of NAD plus BAVL, a molecule crucial for mitochondrial energy production and repair pathways. Supplements aiming to do that are quite popular now.
So lots of different angles. But you said earlier they're all interconnected.
And that's the key takeaway for interventions too. Targeting just one hallmark in isolation probably won't be the magic bullet, because they're all linked in this complex network.
So the future is combination therapy.
That's what the research strongly suggests. The most effective strategies will likely be combinatorial interventions hitting multiple hallmarks at once, synergistically, maybe combining a senolytic with something that boosts mitochondrial function alongside lifestyle changes. That's likely where the field is heading for significant impact.
Okay, so we've really covered a lot of ground here on the hallmarks of aging. What seems like just time passing is actually this incredibly complex dance of cellular changes.
It really is, and so many of these processes are double-edged swords, aren't they? Essential when we're young, but potentially causing problems as we age.
And they're all tangled together. You pull on one thread, like mitochondria.
And you find it's connected to inflammation, to senescence, to telomeres. It's a true network.
So here's a final thought to leave you with.
Yeah.
If our bodies have these intricate, often paradoxical mechanisms built for protection and adaptation, what role do we play? How much responsibility do we have in nurturing those adaptive systems throughout our lives?
Yeah, it makes you think, doesn't it? How do our daily choices, what we eat, how much we move, how we manage stress, how do those choices subtly nudge these deep biological processes towards sustained health span, or maybe unintentionally, towards dysfunction and disease?
It really grounds this complex science in our everyday lives. We hope this deep dive sparks some new connections for you, maybe raise some new questions.
And encourages you to keep exploring this fascinating, rapidly evolving field. Thanks for joining us.